Right. Sorry about that delay, people. We'll start the discussion. We have BioEve paper to discuss, BioEve paper 23, BioEve paper 23, right? And then people, last time there is a statement comprehensive analysis of MCQ that is also there, right? So shall we start with the statement analysis first? Statement analysis. Find that cute people. Okay, I'll give you the chance to mark now. Comprehensive analysis 10.1. Can you mark whether they are correct or wrong? Can you annotate, people? First one, all land plants exhibit alternation of generation. What do you say about that? Is it correct or wrong? Come on, mark. Lavai, can you mark? Can you mark on the screen? All land plants exhibit alternation of generation. Please mark. People, can you hear me clearly? Can you hear me? Can you hear me? At least answer that. Can you hear me? Yes. And then, yeah, okay, good. Then, can you mark on this paper then? You can annotate. Can you mark on the paper? Okay, mark. Come on. Come on, come on, come on. Come on, people. Mark. What's wrong? Shy. Huh? What's the problem? Don't want to mark. What's wrong? What's wrong with you all? Right. So first one, all land plants exhibit alternation of generation. All land plants exhibit alternation of generation. Fine. All land plants exhibit alternation of generation. Let me try my just this exposure. Okay. All right. Right. First one, all land plants exhibit alternation of generation. Correct or wrong, people? All land plants are showing alternation of generation. That is a correct statement. That is a correct statement. Right. Right. All land plants. You have to answer them now. Huh? All land plants exhibit alternation of generation. Correct or wrong, people? Correct statement. Right. Second one, the two multicellular body forms that alternate in life cycle of land plants are the diploid gametophyte and haploid sporophyte, which are morphologically different, therefore called as heteromorphic alternation of generation. The two multicellular body forms of alternation in life cycle of land plant are the diploid gametophyte, wrong, right? We know people in the plant life cycle, there are two stages present. What are those two stages? One is the sporophyte. This is the sporophyte, right? This is sporophyte. That's the sporophyte. And what is the other stage? The gametophyte. This other stage is the gametophyte. This is gametophyte, ne? So gametophyte becomes sporophyte, sporophyte becomes gametophyte. So they alternate in the life cycle. So what do you call this? Alternation of generation. This is alternation of generation, right? Alternation of generation. So they alternately grow. So what happened, people, in the sporophyte? What happened? Sporophyte produce sporangia. Sporophyte produce sporangia. And then inside the sporangia, we can see, and inside the sporangium, we can see people. Inside the sporangium, these four mother cells present. Spore mother cells undergo meiosis and then produce spores, right? The two multicellular body forms. So in this question, it says the two multicellular body forms, the diploid gametophyte. Diploid gametophyte, wrong, no? This one is gametophyte. Gametophyte. This is haploid. This is the sporophyte. Sporophyte. Sporophyte is 2N. So this statement is wrong. Right? This statement is wrong. All right. Next one. Gametophyte produce gametes by meiosis. Gametophytes produce gametes by meiosis. Now we know people gametophyte is haploid. This haploid one can't produce gametes by meiosis. So what happened here? They produce it by mitosis. Right? They produce it by mitosis. So these gametes are going to be formed by mitosis. Right? Gametes are formed by mitosis. So we'll take it as mitosis. Right? No need to mark that. Then all land plants carry out external fertilization to prevent desiccation of gametes. All land plants carry out internal fertilization to prevent desiccation of gametes. All land plants carry out internal fertilization to prevent desiccation of gametes. That is correct. All land plants are showing gametes. All land plants are carried out internal fertilization. Correct statement. Right? All land plants, fertilization is going to be internal. Correct. So fourth statement is correct. Then fifth one, people. Fifth one, female gamete egg is retained in the archegonium and the male gamete antherozoids are released from the antheridium in external fertilization. Right? Female gamete is retained in the archegonium and the male gamete antherozoid. Male gamete, sperm, the male gamete released from the antheridium. Are released from the antheridium in external fertilization. No, plants are not showing external fertilization, right? Now, true that people here in this gametophyte, the archegonia are present somewhere here, antheridia are present here. Antheridia release sperms, right? These sperms are swimming into archegonium during the fertilization, right? So the sperm is released into outer water. The sperm now swims into the archegonium. So what do you call this fertilization? Internal fertilization. Internal fertilization. Right? Always plants are showing internal fertilization. Is that clear? Right? So therefore you can't take this as an external fertilization. The statement says here, the female gamete egg is retained in the archegonium and the male gamete are released outside. So that is an external fertilization. Wrong. Plants always shows internal fertilization. right? They show internal fertilization. Next, so fifth statement is wrong. Fifth statement wrong. Question number 6, seedless plants depends on external water for fertilization, but seed plants do not depending on external water for fertilization. That is correct. I can agree with that. Now, some of the seed plants, if you take the seed plants are using internal water. They don't want rainwater. They don't want external water for fertilization. Right? So, they use internal water. Right? So, therefore, this statement, seedless plants depends on external water for fertilization, correct. Now, here the sperm use external water and then carry out what people? Internal fertilization. So, what is this sperm? Carry out external water. External water is used for internal fertilization. For internal fertilization. External water used for internal fertilization. All right. Okay. Next one. So, I'm taking sixth statement correct. Sixth one correct. Right. Sixth statement correct. Question number 7, statement 7, delay of meiosis after fertilization result in creating a diploid sporophytic generation. Delay of meiosis after fertilization result diploid sporophytic generation, correct. 7th statement, right, correct. 8.1, in evolution, gametophyte generation gradually reduces and has become dependent on the sporophytic generation in seed plants. In evolution, we learn people gametophytic reduction continuously occur, right? So, 8th statement is a correct one. Sporophyte become more and more prominent and the gametophyte becomes smaller and smaller and gradually reduce and become dependent on the sporophytic tissues. That is correct, right? Right. Statement 9. Statement 9. Pogonatum gametophyte is photosynthetic stem, leaf and roots are present in gametophyte. Pogonatum gametophyte is photosynthetic. Pogonatum gametophyte is photosynthetic. Stem, leaves and roots. No roots neither. Rhizoids. Wrong statement. Wrong statement. 9. Wrong statement. 10th one. Pogonatum embryo is retained within the archegonium and developed into diploid sporophyte by obtaining nutrients from the nucellus. Wrong neither. Where is nucellus in Pogonatum? Wrong ne. No nucellus for Pogonatum. Question number 11, statement 11. In Nephrolepis, sporophyte stomata are developed in aerial parts of gaseous exchange. In Nephrolepis, sporophyte stomata are developed in aerial part for gaseous exchange. Nephrolepis people, we learn stomata are present in the capsule. Nephrolepis, sporophyte stomata are developed in aerial parts, right? Sorry, I made a mistake. Nephrolepis, sporophyte stomata present. Capsule people, pogonatum. Pogonatum capsule has stomata, but it's a sporophyte part. Now, this is about people Nephrolepis. So, Nephrolepis, sporophyte stomata are developed an aerial part for gaseous exchange. Correct? 11th statement, right? The stomata present for gaseous exchange. I can agree with that. Pogonatum gametophyte is small heart shape. Pogonatum gametophyte? Pogonatum? Wrong. Right? 12th one, Pogonatum gametophyte heart shape. Who says that? Wrong. Question number 13, Nephrolepis motile sperms swim through external water towards the egg, entering the archegonium in response to chemical attractants. Yeah, correct. Correct. 13th statement, correct. 14th one, in Nephrolepis, after fertilization, zygote develop into the embryo and then to the young sporophyte while retained in the gametophyte. In Nephrolepis, after fertilization, zygote develops into the embryo and then the young sporophyte while retained in the gametophyte. Correct, Ned? Right? Correct statement. Do you understand that? In Nephrolepis, after fertilization, zygote develops into the embryo and then the zygote is going to retain in the gametophyte, correct me? Right? Right. 14th, 15th one, in Nephrolepis, strobili are covered by indusium. In Nephrolepis, strobili are covered by indusium. Nephrolepis, strobili, Nephrolepis, strobili. Wrong, neither. Right? 15. No strobili in Nephrolepis. Wrong. Selaginella, heterophyllous leaves. Heterophyllous leaves arranged in as pairs in strobilus. Wrong. Heterophyllous leaf of Selaginella is present on the stem. The ventral leaf and the dorsal leaf we take it as the heterophyllous leaf. 16 statement wrong. 17 statement, in Selaginella, megasporophyll produce a single megasporangium. In Selaginella, megasporangium produce single megasporangium and microsporophylls produce a single microsporangium. In Selaginella, megasporophyll produces single megasporangium. Megasporophyll produces single megasporangium. Correct, neither. And microsporophylls produce single, yes. In Selaginella, we know people, each leaf has a single sporangium. 70 statement is correct. 18th statement, in Selaginella, genetically two different types of spores are produced. In Selaginella, genetically two types of spores are produced. This nature is called heterospory. No, size-wise different. Wrong statement, right? Not genetically. Statement 19, Selaginella megaspore starts to develop into female gametophyte. Statement 19, check that part. Selaginella megaspore starts to develop into female gametophyte in the strobilus. Megaspore start to develop into female gametophyte in the strobilus and are released into the external environment. Correct, people. We know the megasporangium, the megaspore begins its germination and then transform into partially developed female gametophyte. So as it disperses, it goes as partially developed female gametophyte. Correct, neither. Statement 20. Statement 20. Selaginella female gametophyte is unicellular. Selaginella female gametophyte is unicellular. Wrong. Selaginella female gametophyte is a multicellular one. Wrong statement. Right? I hope you remembered these theory parts. I want you to study people plant life cycles again and again and again and again. So all my questions, right, either BioEve, the CAM paper, right, is going to be mainly focused on those tough parts that you're going to give up, right? So I don't think you can give up it so easily. So, right, when time to come, sir, why is the 16th statement wrong? 16th statement says Selaginella heterophyllous leaves are arranged in pairs in the strobilus, not in the strobilus, right? Heterophyllous. Heterosporophylls are there in the strobilus. Heterophylls are the ventral leaves and the dorsal leaves present on the stem. Okay? Let me show. Selaginella. What do you call that? Heterophylous condition? Am I? Me? Selaginella on the stem, ventral leaves, these are large ventral leaves, and small dorsal leaves are there. This is heterophylous. This condition is heterophylous condition. Now, heterosporophyll is a different condition where you see that in the strobilus. Now, this is the strobilus, right, people? This is strobilus. We don't talk about this. Here, this is strobilus. This is the stem. This one is strobilus. That is strobilus. Right. So we are not talking about strobilus. We are talking about stem. 16. Okay, agreed. Right. All right. Hurry. So we are moving people to the paper now. BioEve paper. Can you take BioEve paper? 23rd paper. BioEve paper I have uploaded to the website. All the paper is uploaded to the website. Paper is uploaded to the website. Right. And then I gave you the printed materials in the class also. Right. Printed materials in the class. I don't know why you are not having the tute yet. Right. After uploading, then after giving it to you, why you don't have it? You must be having it by now. So I will discuss these questions. We'll discuss these questions in the BioEve paper 23. Paper 23. Right. Paper 23. First question. The first form organisms on Earth. First form organisms on Earth. Now you can, those who don't have the tube, they can take it, right? Sir, why they say sporophylls are compactly arranged in the strobilus? Sporophylls, spore producing leaves, are tightly packed so that they make the strobilus. Simple. Otherwise, right? Otherwise, they are compact. Strobilus, they are compact. You saw that, so they are present compact in the strobilus. So the compact, right? You saw that here. See the strobilus. So you can see that they are compact. Fields are compact, right? The megaspore fields, micro. The color thing is micro. The green color stuff there, the megaspore. So compact. So this is the microspores are dispersed. These are the megaspores. Look at the size of the megaspore. Large megaspore, right? Big megaspore. All right. Okay. First question, BioEve 23. First question, paper 23. Paper 23. BioEve. First question. Right. First question. The first form organisms on Earth was formed about 3.8 billion years ago. 3.8 billion, wrong, neither 3.5 billion years ago. And then it was arose in the primitive soup. Yeah, correct. Was anaerobic prokaryotes, correct. Correct. Regarding the above, I'm taking second one, B and C only. B and C. My answer is second. Second is the answer. Question number 2, which of the following correct regarding water? It is straight. It is a straight polar molecule. No, angular, polar, wrong. Addition is the attraction found between water molecules due to hydrogen bonds. Addition, attraction between, no. Water and some other molecules. Wrong. Water is high surface tension due to addition between water molecules. Water has high surface tension due to addition, wrong, cohesion. And then water has maximum density at 4 degrees Celsius, perfect. Fourth one is a correct one. substance can dissolve in water regarding its ionic nature? No. Water, substances can dissolve in water regarding its ionic nature. Now, water ionic nature is not helpful in solubility. Now, we learn substance ionic nature can be helpful. Now, sodium chloride is ionic compound and its ionic nature can be helpful to be solubilized, right? Right? To be a good solvent. But water, sorry, good solute. But water is a good solvent because of its polarity. You understand that, ne? Right. So my answer is fourth one. Question number 3, which of the following statement is correct? Adenosine is a nucleotide which produces ATP. Adenosine is a nucleotide, wrong. It is adenosine is a nucleoside, right? Adenosine is a nucleoside, wrong. Then NADP plus function as oxidizing agent. NADP plus. Now look at this. NADP plus, right? NADP plus. NADP plus. NADP plus. NADP plus, it can become NADPH. NADPH. That means it absorbs hydrogen. It takes hydrogen. It can take electrons. Electrons also. When something absorbs hydrogen and electrons, this is a reduction reaction. This is reduction. It is reduction. Right? So what happened? When NADP plus become NADH, it undergo reduction. It undergo reduction. So someone has to give electrons. Let's take X is donating electrons and H plus to transform into something else. So what do you think happened for X? Right? X is now giving H plus and electrons for this NADP plus to become NADH. So what happened to the X? X become plus, right? Because it remove electrons. So X undergo oxidation. X undergo oxidation. X undergo oxidation while NADP plus undergo reduction. Therefore, NADP plus is an oxidizing agent. He himself undergo reduction. That is not what we want. What he does on the other. So what is NADP plus? NADP plus is an oxidizing agent. I think you understood this. Right, Lamai? Teruna neda meka. Harida, simple neda. Simple neda. Oh, simple. Simple neda. Hari. Ilangada, question number statement 2, NADP plus function as an oxidizing agent, correct? RNA is always single-stranded nucleic acid. No, it sometimes can be double-stranded. And mRNA is an abundant RNA type in the cell. mRNA is an abundant RNA type in the cell. mRNA, no. What is most abundant? Ribosomal RNA, right? And what is the largest one, people? What is the smallest tRNA? Smallest is tRNA. That's given in the resource book. GTP is a nucleotide and can provide energy, right? GTP is a nucleotide and can provide energy. GTP is a nucleotide. It is a nucleotide, correct, and can provide energy. Some events we can see that GTP supply energy, but you have not learned that part yet. So out of these answers, people, I would like to go with the second one as the best. Third question, I'll go with the second. This last statement, the fifth one is not wrong. GTP is a nucleotide, but it's not a simple nucleotide. It's a nucleotide with, right, guanosine triphosphate. GTP means guanosine triphosphate, right? Guanosine triphosphate, like adenosine triphosphate. Guanosine triphosphate is energy donor in some reactions, right? but you can learn later this can provide energy, right, in DNA inside the nucleus, right, some events it can provide energy. GTP is a nucleotide and can provide energy. I don't refuse it completely. It is a nucleotide with 3 phosphate groups, right? Correct. So I'll take that third one, fifth one also, right? Second and fifth. Question number 4, synthesis of glycolipid. Transport protein synthesized by bound ribosomes. Functions of both endoplasmic reticulum. What do you take, people? Functions of endoplasmic reticulum. Can somebody send me the answer? Synthesis of glycolipids? No. Neither. Transport of proteins synthesized by bound ribosomes? Endoplasmic reticulum? Yes. Transport of proteins synthesized by bound ribosomes. Functions of both endoplasmic reticulum. They are asking SCR, RER, both functions. Ne? So we can't take them. Then produce transport vesicles done by both SCR and RER, correct. And then synthesis of glycoprotein. Glycoprotein, no. That's done by RER. Ne? And carbohydrate metabolism, no. Functions of both endoplasmic reticulum. C, D, E, synthesis of, no, ne? C only. Second one, ne? C only. Lamai, second one. I don't know, I have marked it as fourth one before, right? I think C only. Synthesis of glycolipids. Do you agree with me? C only. B and C. One of you say B and C. Transfer of protein synthesized by bound ribosome. This is done by RER only. Right? Only C. My answer is second there. Right? Answer is second, okay? Question number 5, people. Correct statement regarding photosynthesis. Question number 5, correct statement regarding photosynthesis. Water splits during linear electron flow without precipitation of enzymes. Water split during the linear electron flow without the precipitation of enzymes. Sorry, without the participation of enzymes. Water split during linear electron flow without participation of enzymes? No. It is an enzyme catalyzed reaction according to the resource book. Wrong. Then 3PGA is a primary product of photosynthesis in all plants. 3PGA is a primary product of photosynthesis in all plants. Phosphoglyceric acid, primary product of photosynthesis in all plants. 3PGA, phosphoglyceric acid. Wrong, neither. Primary product of photosynthesis in all plants. Phosphoglyceric acid is produced, right? Phosphoglyceric acid is produced in C3 plants. First table, first product, neither. But in C4 plants, oxaloacetic acid. 3PG is a primary product of photosynthesis. Wrong, ne? We'll take it as refuse it. Then people, electrons from splitting of water are integrated into NADP plus. Correct, neither. If electrons flow in linear electron flow, these electrons are going to end up with NADP plus, forming NADPH. Correct. Rubisco is only involved in the fixation of carbon dioxide. Rubisco is only involved in the fixation of carbon dioxide. Rubisco is only involved in fixation of carbon dioxide. Rubisco. No, Rubisco is having the ability of fixing oxygen too, neither. So it carries out carboxylation as well as oxygenation reaction. Wrong statement, Lamai. Then malate is more stable compound than that of the oxaloacetate. Malate is more stable compound than oxaloacetate. What do you say about that, people? Is it given? Is it given? Can you check the resource book for that? I think there is a statement to support that. Right? Fifth one, malate is more stable compound than oxaloacetate. Can you check that? Electrons, can you check that in the resource book, people? I think there is a statement for this. You have to check photosynthesis lesson, C4 pathway, photosynthesis. In photo PGA, C4 plants. In most C4 plants, expand physiology, the bundle sheath cells enclose vascular bundles. Oxaloacetate is rapidly converted into more stable C4 acid malate. Right? Yes, so there is a statement. It says that. So we take two answers there. Electron form splitting of water integrate into NADP plus 4 and 5. 5, 3 and 5. Right? 3 and 5. Malate is more stable compound than that of oxaloacetate, correct? Electrons from splitting of water integrate into NADP plus. That is also correct, neither. It can integrate into NADP plus, yes. So, isn't the electrons from the splitting of water used to neutralize the excited PS2? Yeah, that's correct. That's correct. Finally, no. Immediately that happens. Immediately it happens. Yes, correct. Shall we check that part? I think then since we have another answer, neither. So check that part, people. PS2 splitting of water. I think there was a statement like that. Immediately going to neutralize it, but then finally we can see that these electrons are on the go. So it says electrons release yield oxygen. Electrons will be accepted by primary electron receptor of photosystem 2. Striking a photon of light pigment, the process is called linear electron flow. The key for transformation, pigment result, these electrons will accept by primary electron receptor of photosystem 2. Linear electron light absorb photosynthetic pigments and synthesize ATP and NDPH due to excitation of photosystem 1 and 2, which are embedded in the thylakoid membrane of the chloroplast. The key transformation, splitting of water take place as a result of an enzyme catalyzed reaction and yield oxygen and H plus. Electron release as a result of hydrolysis may neutralize excited photosystem 2. Right. So we'll take then people best answer. We'll go with the fifth one. We'll go for the fifth one as the answer. Right. Fifth is better. Fifth one is better. Question number 6. The select, I think, select correct, na? Select, na? Select correct one regarding Krebs cycle. One molecule of ATP is produced during this cycle by oxidative phosphorylation. One molecule of ATP is produced during this cycle by oxidative phosphorylation, Krebs cycle. One molecule of ATP is produced during this cycle by oxidative phosphorylation. Wrong, neither. Substrate level phosphorylation. First statement is wrong. And aerobic respiration, much amount of carbon dioxide is released in this cycle. In aerobic respiration, much amount of carbon dioxide is released in this cycle. Yeah, how many carbon dioxide come, people? 2 carbon dioxide is released for every pyruvate. Second is okay. 6 NADH and 2 FADH2 are produced in this cycle. 6 NADH for one glucose molecule, then it is correct. 6 NADH and 2 FADH2 are produced in this cycle. Krebs cycle. When the cycle run twice, it can happen. Right? And then NAD plus is reduced into NADH using energy by breaking of glucose. Wrong, neither. NAD plus is reduced to NADH using energy by breakdown of glucose. And then citric acid cycle is produced as the primary product. Citric acid cycle is produced as the primary product in this cycle regarding carboxylation. Citric acid is produced as the primary product in this cycle. In this cycle regarding carboxylation. I think the second statement is more valid, right? We'll go for the second one. Select the correct statement regarding Krebs cycle. In aerobic respiration, much of the carbon dioxide is released in this cycle. Krebs cycle, correct. Other statements are not very clear in their idea, isn't it? So we'll go for the second one. Question number 7. If you have arguments, send me. So I did not understand the statement. Last statement. Citric acid is produced as primary product in this cycle regarding carboxylation. It's not clear. It's not clear. We'll just leave that. Then what about the third one, Kilhano? Third one, 6-NADH and 2-FADH2 are produced in this cycle. No, Krebs cycle when it is run twice. Right? So third can be also be taken. No, third can be taken if it is given for one glucose. Otherwise, you can't take one cycle, produce how many? 3-NADH, 1-FADH2. Yeah, that's the issue. So we'll go with the second one. Question number 7. Common characteristics of both domain archaea and eukarya. Common characteristics of both domain archaea and eukarya. I'm taking people fifth one as the answer. That is easy, neither. Fifth one as the answer. Peptidoglycan thing. Question number 8. Select the correct answer regarding hierarchy of taxa. Mammalia is vertebrate phylum level. Mammalia is a class, wrong. Moving to next page, Lamai. Crustacea is the invertebrate order level. No, it's not Crustacea. Right? It's a class 2. And then Zelanicus is the species level. It's the Zelanicus specific epithet. Neither wrong. Panthera pardus is species level, correct. From domain to species, number of similar characters among the members in the taxa increases. Domain to species, number of similar characters, the common characters, neither, not similar characters. So we are taking people the answer, fourth one for question number 8. Similar characters? No, common characters. Question number 9, which one of the characteristics of chlorophytes, which were the ancestors of land plants? Which were the ancestors of land plants? Walled spores produce sporangia, chlorophyll AB pigments, dependent embryo, and apical meristem, multicellular gametangia. Check those points, people. One of the characteristics of chlorophytes which were ancestor of land plants. How do you say that the land plants and chlorophytes are related? I'm taking people presence of chlorophyll AB pigments. Second one, I think we discuss. Yeah, we discussed this, having cell wall, cellulose cell wall, starch, and then having these pigments are making a relationship. Question number 10. Corrects regarding responses of the plants which they get stimuli from the environment. Canopy of the forest absorb far red light rather than the red light. Wrong. Canopy absorb red light, not far red. Wrong. Right? Canopy absorb red light. So what comes down to the plant under the shade? The far red. So this is far red, therefore can result some photomorphogenesis. This is the forest. This is the canopy, right? When red light fall, it absorbs for photosynthesis. What comes down is far red, far red, far red, isn't it? Comes far red down. So canopy of the forest absorb far red light, wrong. Red light inhibits seed germination, wrong. Far red inhibits. And then exposure to direct sunlight allow the plant to grow tall, wrong. Branch. Statolith play a major role in negative geotrophism. Statolith play a major role in negative positive geotrophism, wrong. Far red light inhibits seed germination in plants. Far red light inhibits seed germination, correct. So my answer is 5. 10th question, answer 5. Right, we'll check the remaining questions. Question number 11. Following are plant growth regulators and their functions. Select the correct combination regarding plant growth regulators. That's a tough question. I think you can answer this. We have to go on checking people the given combination for this sort of a question, right? The given combinations you check, all right? And Balanda, first one, AR, auxin and stimulate stem elongation, correct? BP, gibberellin and inhibit growth, no. So first we reject. Second one, AP, auxin inhibit growth, wrong. Third one, AS, auxin enhance apical meristems, correct. BR, gibberellins are stimulate stem elongation. Stimulate stem elongation, correct. And then CT, cytokinin T, stimulate seed germination, correct. DP, abscisic acid P, inhibit growth, correct. EQ, ethylene Q, promote leaf abscission, correct. So third one is perfect. AR, BR, how about that BR? Auxin stimulates stem elongation, correct. Neither. And then CP, cytokinin inhibit growth, wrong. So fourth one you can reject. Fifth one, AS, auxin S, enhance apical meristem, correct. BT, gibberellin T, stimulates seed germination, correct. CR, C, cytokinin, R, stimulates stem elongation, no. So you have the answer, 3. 11th question, 3. 12th question, deficient elements to plants that show wilting. Deficient elements to plants that show wilting. Hydrogen and what else? Hydrogen and chlorine. Result deficiency, what do you call desiccation, wilting. So I'm taking third one as the answer. 12th question, third is the answer. Question number 13, photosynthesis reduced and short-lived gametophyte found in. Photosynthetic, reduced and short-lived gametophytes are present in Pterophyta. I'm taking 5 as the answer. Think you agree with me. Pterophyta, photosynthetic, reduced and short-lived gametophyte is there. Bryophyta don't take that because they have a long-lived gametophyte. Question number 14. Question number 14, cholenchyma cells are found in vascular plants. Yeah, all vascular plants, wrong. Neither. It says all vascular plants. No. Cholenchyma not present in all vascular plants. No. And then provide mechanical support in roots, wrong. Completely absent in root system, cholenchyma. Elongate with stems and leaves, they mechanically support, correct. Third is a perfect one. And concern of corners of cell walls are unequally thickened with lignin. Have corners of the cell walls are unequally thickened with lignin. How about that, people? have corners of cell walls are unequally thickened with lignin? No, no lignification there. And have only primary walls and walls and are spherical. No, they are not spherical in shape. Primary walls only, correct. But I have the answer now. Third one is my answer. I think you agree with me. Question number 15. Influx of water, increase of water potential, bending of inner wall, increasing of turgidity. Now they are asking what is the right order for opening of stomata. What's the correct order to open a stoma? This is a guard cell. This is a guard cell. What happens, people? This guard cell, in presence of blue light, when blue light present, in presence of light, we'll just take it as light, neither. In presence of light, right? When light present, energy means ATP present, so that it actively take potassium ions in. Right? The active absorption of potassium ions in is now helping in transport of water. This makes the cells turgid. That is now making the cell turgid. And as a result, these walls now bend unevenly, opening stoma. So, correct sequence of the events. Decrease of water potential. Why water potential decrease? Potassium transport occur. So, water potential decrease. Water enter. So, we will take decrease of water potential first. Then influx of water second. Right? And then the increasing in the turgidity. And then we will take people expansion of the cells. And finally, bending of the inner wall. I'm taking fourth one as the answer. Question number 15, fourth is the answer. Right, next question. Next question. Next question, question number 16, correct statement regarding bones. I'm taking people fifth one as the answer. Central canal is found at the center of the osteon containing blood vessels and nerves, correct. Canals are found at the edges of the osteon. Connective tissue which contain less minerals. Matrix is only made up of collagen fibers. And then specialized connective tissue. Specialized. I think this is mineralized. That is the word they have used. So we have the best answer for 16th. 5, I think, gives a better description. Then fibers in human food. Fibers in human food. These fibers are the plant fibers, right? Contribute to protect against cancers in the colon, correct. 17, my answer is first, right? Are made up of polysaccharide and fatty acid? No. And are used to synthesize fibrous connective tissue? No. Aid in absorption of calcium? No. Act as a carbon source needed for metabolism? No. Question number 18, correct response, responses regarding cofactors. They are non-proteinous components, correct. All are organic compounds, cofactors, inorganic cofactors are there too, wrong. They can be bind temporarily with enzyme molecule. Yes, correct. And they can bind permanently with enzyme molecules. Yes, some bind permanently, some bind temporarily. Yes, they always need enzyme activities. They always need two enzyme activities. Cofactors not always. Name what they have given in the resource book. Sometimes, most, sometimes. For some enzymes. So I'm taking ACD. ACD as the answer for question number 18. ACD. Moving to next page, we will question number 19. Following are some characteristics and protist. The correct combinations regarding character and the protist. Correct combinations. What are you going to take, people? Select the correct combination. PZ. Cellulose cell wall Z. Ulva. Cellulose in cell wall ulva. Correct Nedalamai. PZ is okay. Then QW. Q multicellular thallus. W sargassum. Yes. RX contractile vacuole X. Amoeba. Correct. SY, chlorophyll C, Y diatom, correct. Then PW, cellulose in the cell wall, sargassum, correct. QZ, multicellular thallus, Z, ulva, correct. RX, contractile vacuole, X people, amoeba, correct. And SW, chlorophyll C and sargassum, SW, chlorophyll C, sargassum, correct. So A, B, correct now. Then C, PY, P, cellulose in the cell wall, Y, diatom, wrong. No cellulose in the cell wall of diatom. Silica present. And then people Q, so PY reject. So I'll drop that. No point of checking the rest of the part. PW, QW, RY. RY we didn't check before. R and Y. Contractile vacuole diatom, wrong. So you can't take D. Then PX we didn't check before. Cellulose in the cell wall, amoeba, wrong. So AB must be the answer. Question number 19. AB is the answer. AB is the answer. Question number 20. Which of the following is correct regarding life cycle of plants? Correct regarding life cycle of plants. Gametophytes of Pogonatum is dioecious and long-lived than they are sporophyte. Gametophyte of Pogonatum is dioecious. Pogonatum. Dioecious means what, people? Two houses, neither. Male one in one house, the female one in other house. What do you call that? Unisexual, ne? Dioecious. Look at this. This is one house. One house. Both sexes, the male, female ones are in the same house. Then what do you call monoecious? Ecious means house. Monoecious. Dioecious means what, people? Two houses. Male in one house, female in another house. Female in another house. So what do you call this? Dioecious. Dioecious, on the other hand, unisexual. Monoecious, on the other hand, bisexual. You learn all these things. So, which of the following is correct regarding life cycle of plants? Gametophyte of Pogonatum is dioecious and long-lived than they are sporophyte, correct. Cuticle is found in the aerial parts of sporophyte of Neplephis, correct. Selaginella bear heterophyllous leaves and ovules, wrong. No ovules in Selaginella, wrong. Right? So sad, look. And then, so sad for what? Monoecious or dioecious? So sad. Huh? So sad for what? Monoecious or dioecious? I say monoecious, so sad. Dioecious, yeah, happy. Right? So, the anthophytes contain highly reduced male gametophyte, which is found within the pollen grain. Anthophytes contain highly reduced male gametophyte, which is found in the pollen grain. Correct, ne, Lamai? Yes. So ABD okay, ne? Seekers need external water for fertilization. Seekers need external water for fertilization. No, they don't want external water for fertilization. Wrong. So ABD, right? ABD. ABD is the right answer for that. Both of them are so sad at that point. This point, ne? Right? This point, ne? She's now ready with the dinner, right? The brew. Ready with the dinner, right? This guy is ready with the bottle, ne? Bottle, right? How about that? Right. Right? Isn't it? Now we'll go for the structured. We have very few structured questions. I'll give you the essay answer, essay scheme I'll give you. We'll quickly finish structured. Right? We'll quickly finish the structured part. Where is your dinner? Already I had my dinner, right? Enough now. Enough of that dinner. Right. Paper BioEve. Right, people, we have to go through this structured questions quickly, right? Structured questions we have to go through quickly. Few questions, ne? We'll do that fast. First one, I'll read. Can I read the answers? Right? No need to write. Ne? It's going to take time if I write them. What is a method of water and solute transport in plants? What are the method of water and solute transport in plants, people? You can say those methods, ne? Diffusion, diffusion, diffusion. What are the other methods? Osmosis, osmosis, imbibition, imbibition. What else? Facilitated diffusion, facilitated diffusion, facilitated diffusion. And last one, people, bulk flow, bulk flow. Right. Write the factors that affect water potential, solute concentration and physical pressure. Physical pressure. And what is the other one? Solute concentration. Solute concentration. Right, next one, what is meant by radial transport? Transport of water and minerals. Transport of water, water and minerals, minerals that entered from soil to the root cortex. Entered into root cortex, into the xylem, entered into root cortex from soil, from soil to the xylem, of root, right? Next one, name 3 routes. Do you know 3 routes, people? Number 1, 2, 3. What are those 3 routes? The apoplastic route, symplastic route, and then transmembrane route. Okay. Give difference between epidermis and endodermis of root. Epidermis, root hairs, right? Then no root hairs. Or maybe you can say no Casparian, no Casparian strip in the epidermis, Casparian strip. You can see Casparian strip present. Strip present. Right, they are asking only one difference. I think you wrote that. Then common life cycle of plant is given. Name A. What is A, people? Gametophyte, ne? Gametophyte. What is B? Sporophyte. Sporophyte. Done that. Moving to next page, my dear students. Next page. Oops. I did a mistake. Oh, sorry. I didn't share it. Ne? Ayyo. What's going on? Ah. All right, it's not difficult. You did that part. All the methods, physical pressure. Okay, no issue. You all are good people. Right. Next one. Next one. Name the plant where A is independent and dominant. I'm taking Pogonatum. Pogonatum. Then I don't have an answer for this one, people. We'll remove it. Neither. Plant where A is monoecious and independent kiwo okay ne? Independent nang okay. Dependent, I don't have an answer. All right. Then part 2, indicate the stage of meiosis with letter X in the life cycle. Where are you going to mark the meiosis? Suppose here. You have to mark here as X. Mark here as X. Right. Then the list of important characteristics of above plants are given below. Select the plants that show these characters. Megaspore released to external environment. What is that, people? Selaginella. Selaginella. Shall I write the letter? I'll write the letter. Selaginella can make the C. C. Plant fiddlehead. Fiddlehead means lamai B. B. And seed surrounded by fruits. Seed surrounded by fruits. E. And xylem tissues without vessels. D. And then people spores produce inside of the capsule. A. Give two features, those found in Selaginella, not in Neplophis. What are you going to say, people? Heterospores. Selaginella, Neplophis, no heterospores. Selaginella strobilus. This one, what do you call Saurus? Neplophis saurus, Okay, you can write something like that. Then, people, this diagram is showing you plant. Identify the above diagram. This is, I'm taking mature ovule. Mature ovule of Anthophyta or flowering plant. Name parts A to H. What is A? A, embryo sac. Embryo sac. What is B? B, integument. What is C? 3 antipodal cells. What is D? D is, what is D? What is D? I think D, ovule. I'll take it as ovule, not clear. Can be nucellus or maybe I'll take ovule. Megasporangium, ovule. E, 2 polar nuclei. 2 polar nuclei. F, F synergids. F synergids. G, I'm taking as egg cell. Okay, all right. Write 3 differences between the above structure and similar structure in Seekers. Above structure and similar structure. So can you show page 3? All right. We'll move to this last part now. Write 3 differences between above structure and similar structure in Seekers. Similar structure in Seekers, what is this? above structure. You can take ovule, the both ovule. Three difference between the above structure and the similar structure in cecus, ovule. Or shall we take the female gametophyte? Embryo sac. Above structure and the similar structure. Now we took it as a mature ovule. Then we'll take the mature ovule, we'll compare. Right? What are you going to say, people? Here, 7 cells in embryo sac. Then no embryo sac. Sac in the ovule of the cecus. Right? What else you going to write? Right? What else? Then you can say archegonia present in cecus. Ne? Oops, I think I wrote it in the wrong side. Ne? So what did I write? Shall we take, okay, archegonia, no archegonia. Archegonia present, ne? Archegonia present. Then one egg. Here several egg cells. Right, this one ovule covered by wall of ovary. No cover. No cover, no wall of ovary. Right. So the next one, people, if you want, you can say stalk present, stalk absent, but that's not the important point. Name structure of following parts after fertilization. B. B becomes seed coat. B becomes seed coat. Then E. What is E? E polar nuclei, the endosperm. F. What are you going to say? F. Antipodal cells disappear. Disappear. So that's it. Now, people, this the essay question. This essay question. I'll give you the marking scheme. Please check your essay answers against this marking scheme. Can you take a screenshot of this page? Put a screenshot at dagan lamai. Can you? Oh, wait, I have to share. Wait, I'm mad. Right, can you take a screenshot? Right, so this is a simple question. Briefly describe the phloem tissue. Type of cells present in the phloem tissue. Second one, briefly describe the mechanism of phloem translocation. I think this is a simple question. I have prepared the scheme. No big difference with the resource book answer also, people. Please keep it ready. I am going to check these things in Saturday class. Right, so time up, and we'll meet soon. Right, next time. Until that, see you all. Bye bye. Have some good time.